Welcome everybody to today's last session. It's a great pleasure to introduce our next speaker, Rachel Freethy. Rachel is an associate professor at the University of Exeter, and she is sort of focusing her time and energy on solving the mystery why a low birth weight is associated with an increased risk of developing type 2 diabetes during adulthood. And you are heading the Early Growth Genetics Consortium, which is probably the effort focused on disentangling how genetic variants impact early growth traits. And I think you're using a number of innovative tools, one of them being Mendelian randomization analyses and several others. And I also wanted to flag that you have sort of an atypical career in the sense that you, after your bachelor studies at the University of Oxford, went to teach at a second grade school for 5 years and then went on to do your master's and your PhD studies. So you clearly not only care about research, but also about scientific outreach and education. So please join me in welcoming Rachel Freethy.
Thank you so much, Tuna, for that very kind introduction. It's a real pleasure to be here to enjoy this fantastic meeting. I've enjoyed and learned a lot from all the talks so far, and it's a real privilege to be able to tell you a little bit about some of the things that we've been doing. So, over the last 30 years, studies of a variety of populations have documented this really interesting association between birth weight and the risk of type 2 diabetes, such that individuals born in the lowest categories of birth weight are more at risk 50 or 60 years later of developing the disease. Now, there have been various potential explanations for this. It's been replicated in a variety of diverse populations, and it really holds up observationally. So the observation led to the proposal of the fetal origins hypothesis. So if the fetus is undernourished in utero, then a direct effect would be reduced fetal growth, but also indirectly via programming or adaptations made by the developing fetus to the surrounding intrauterine environment. This might be expected to lead to constraints that lead to an increased risk of diabetes that become shown later in life. Now there have been there's been support, lots of support for this model from animal models and from human studies, especially humans exposed to famine. But alternatively, the association may have an underlying genetic explanation. So individuals may inherit variants of genes which influence insulin secretion or insulin action, and those would be expected to lead directly to an increased risk of type 2 diabetes. But also, since fetal insulin is a key fetal growth factor, those same genetic variants may be expected to have a phenotype earlier in life as well. So under this hypothesis, the fetal insulin hypothesis, type 2 diabetes and lower birth weight are two phenotypes of the same genotype. Now, these two hypotheses are not mutually exclusive, but I want to focus on the evidence and kind of where we are today in understanding the contribution of the fetal insulin hypothesis. So it's worth going right back to kind of the evidence that led to the proposal of this hypothesis, and this was from studies of monogenic diabetes. So the evidence on which this hypothesis was based was from a study of rare mutations in the glucokinase gene. Now, glucokinase is the pancreatic glucose sensor. It catalyzes the phosphorylation of glucose to glucose 6-phosphate, which is the rate-determining step of glucose metabolism in the pancreas and in the liver. And individuals who inherit heterozygous glucokinase mutations have a defect in glucose sensing in the pancreatic beta cell, which means that the glucose has to reach a higher set point before insulin is produced. And that means that people who have heterozygous glucokinase mutations have a mild, stable, fasting hyperglycemia throughout life. But also, as this data shows, in a study of sibling pairs who either did or didn't have a glucokinase mutation, it also leads to reduced birth weight. And that's because there needs to be more maternal glucose, a higher level of maternal glucose before the fetus produces insulin in response. And then as fetal insulin is a key growth factor, that makes the fetus grow. And actually there's quite a big difference in birth weight between individuals who do and don't inherit glucokinase mutations. Now, just how important fetal insulin is in human birth weight has been really nicely demonstrated in another study of monogenic forms of diabetes, just published this year, actually. And this work was led by Alice Hughes, and it's actually the first time I've presented this work. And so what Alice did was to look at the birth weights of babies who inherited recessive loss-of-function mutations in the insulin gene or were born with pancreatic agenesis. So these babies have no fetal insulin. And what you can see when you compare their birth weights to the kind of standard normal growth curve based on the intergrowth data is that at term, at 40 weeks, as you can see here, the birth weight is about half of human birth weight. And that's actually, so insulin-mediated growth accounts for a higher proportion of human birth weight than any other species. The other thing you can see from the data is that, and this is about, so this is kind of 30 weeks of gestation, the difference produced by insulin-mediated growth becomes apparent only in the kind of last trimester of pregnancy. So these rare monogenic mutations establish this principle that so lower fetal growth and diabetes can be two phenotypes of the same genotype. The other thing going back to glucokinase mutations, the other thing they do is show really nicely that it's important in any genetic studies of a perinatal phenotype, such as birth weight, it's really important to consider that both maternal and fetal genetics may have independent effects. And so we need to account for those in our studies. So if we look now at mother-child pairs who either do or don't have glucokinase mutations, if you have, the mother has the mutation, but the baby doesn't, then the mother's glucose is at a higher set point, and the baby will produce more insulin in response to that mother's glucose, and the baby will grow bigger. If the baby has the mutation, but the mother doesn't, so the baby's inherited it from the father, then the baby's growth will be reduced due to lower insulin secretion. But if both mother and baby have the mutation, then those two opposing effects cancel each other out, and the baby is born with a birth weight that's no different from average. So both fetal and maternal diabetes genes can influence birth weight. But of course, these rare mutations, although they helpfully establish this principle of a link between birth weight and diabetes, because they're rare, they can't explain those associations that we see in the general population. And so what's needed are examples of common variants that influence both birth weight, normal variation in birth weight and risk of type 2 diabetes. And that's what we've been looking for. And over the years, I've been involved in some really great collaborations, which have resulted in successively larger genome-wide association studies of birth weight. And we've looked at these and we've compared them with genetic data on type 2 diabetes. This data is from our most recently published GWAS of birth weight, which was published in 2019. And the results are summarized here. So each point on this plot is the association between a common SNP, a common variant that we've studied, and birth weight. The results of the fetal genome-wide association study, so an individual's own DNA and their own birth weight plotted going up here on the y-axis, and the associations with birth weight in the maternal study, so maternal genetics and offspring birth weight plotted going down. And then genomic position, if you laid all the chromosomes end to end, is on the x-axis here. And what this kind of shows in summary is that we have a large number of nearly 200 regions of the genome where there were genetic variants were robustly associated with birth weight. But what we did see, of course, and this is not a surprise, since the maternal and fetal genotypes are 50% correlated, there were several signals that came up in both the maternal and the fetal study. And to get any further to understand these results, what we need to do is to be able to say, well, are these signals coming from the the baby's DNA and they're just tagged by the mother because they're correlated, or vice versa, or are there contributions from both mother and baby, as we saw with there could be with the glucokinase example. So just to illustrate this, the baby will inherit genes from the mother and the father, and those genetic variants in the baby may have a direct effect on birth weight, but also maternal genetic variants, regardless of whether they're passed to the baby, 50% will be, regardless of whether they're passed to the baby, may impact the intrauterine environment and thereby influence birth weight that way. And that's what I mean when I say we have to account for the correlation between. We have to make sure we can adjust the one for the other. Maternal genetics may influence, for example, the level of glucose available to the baby, the blood pressure, the level of other metabolites. And so we need to take those into account, which for quite a long time actually presented quite a problem, because in order to get the power to identify the effects of common variants, which are small, we need hundreds of thousands of individuals in a well-powered genome-wide association study. And we can combine samples together, but we don't have a comparable number of parent-offspring trios in which to dissect those effects. So within the Early Growth Genetics Consortium, in our last GWAS, we had 300,000 in the fetal GWAS, 200,000 in the maternal GWAS, and we have about 70,000 father, mother, child trios with GWAS data available. But of course, that's an order of magnitude lower in power, so we can't use those in conditional analyses to dissect the effects. But that's what's wonderful about bringing collaborations together where we can share data, is we also bring expertise together. And so it was great to work with Nicole Warrington and Dave Evans from the University of Queensland with their expertise in statistical genetics. And they worked out that where we have a study such as the UK Biobank, where you have an individual's own DNA and information on their own birth weight, and also information on their offspring birth weight, you can use a structural equation modelling approach, which can incorporate the data from a fetal and a maternal GWAS and estimate the conditional maternal and fetal effects on birth weight without needing a parent-offspring trio. And they've shown that this can work really well. It's very flexible. You can incorporate summary data from really large genome-wide association studies. And so that enabled us to take our SNPs that were associated with birth weight in our study and say for some of them, there was only evidence of a fetal effect. So, for example, the HMG02 locus, which is very well known to be associated with human adult height, also has an influence on birth weight, probably because it's influencing length right from the word go. There were several variants where we saw only a maternal effect. So, for example, MTNR1B, which is a well-known locus associated with fasting glucose levels, that has an influence on birth weight when present in the mother. And then for others, we found evidence of loci where there were maternal and fetal effects, independent effects, either in the same direction or in opposite directions. Since that study, it was wonderful to see the Decode Genetics group produce an even bigger GWAS of birth weight, where they meta-analysed the data that from our study with their own data, including consideration of birth length and including a paternal GWAS. And what's wonderful about the Iceland population is that the Decode study is that they have information which enables them to look at phased haplotype from the extensive pedigree information. And they were able to look at comparing maternal and paternal transmitted alleles with non-transmitted alleles. So as well as increasing the number of genetic loci associated with birth weight, they were able to look at maternal effects, fetal effects, and also they were able to identify parent of origin effects, where the effect on birth weight depends on whether the allele is inherited from the mother or from the father. So that was great. Okay, but back to our question. Do type 2 diabetes risk alleles reduce birth weight if inherited by the fetus. So overall, there's definitely evidence that they do. So this is a plot which shows a genome-wide genetic correlation between birth weight, this is on the y-axis, and a range of different other phenotypes. And I've just circled the glucose-related phenotypes here. So type 2 diabetes, indices of glucose levels or fasting insulin, and the associations with triangles are the maternal association, the correlations between these traits and the maternal effects on birth weight. And then the circles are the fetal effects on birth weight. And as you can see, the higher risk of type 2 diabetes in the mother or higher risk of high glucose is positively associated with birth weight, whereas in the fetus we see the opposite. We see an inverse association with birth weight, which mirrors what we saw in the rare glucokinase examples. So genome-wide, overall, we see these genetic correlations in the direction that we would expect under the hypothesis. And then when we look at several individual loci, so this is a variant in ADCY5, which is known to be associated with type 2 diabetes risk. The type 2 diabetes risk allele is associated with lower birth weight, and also from recent studies that we've done with a lower level of umbilical cord insulin. And so that all fits with the hypothesis. So are all type 2 diabetes risk allele risk variants associated with lower birth weight? Well, overall, the answer to that is no. So what I've got here is a number of genetic variants or loci that are known to associate with type 2 diabetes risk, which have been classified using information on their associations with other traits into their likely mechanism of action. In terms of type 2 diabetes risk. So we have those that influence beta cell function, proinsulin processing, those that predispose to type 2 diabetes via a primary effect on obesity risk, and then a group which have a lipodystrophy-like phenotype, so they're concerned with adipose tissue distribution and insulin resistance, and then a group associated with liver function. What we've got on the x-axis here is the there we go. I lost it for a minute there. The fetal effect on birth weight. This is the 0 effect line. And so what you can see is many of those genetic variants that we know to associate with type 2 diabetes are not associated with birth weight. A number of them are, and overall those that are show associations with lower birth weight, so that's in the direction we'd expect. So a couple of points to make from this. One is that many of them don't associate with birth weight, and so that actually gives us a kind of a clue as to when in the life course they might be acting. So those that do associate with birth weight, we can hypothesize that they're influencing insulin secretion or action at a very early stage, whereas others, so for example, TCF7L2 with the largest effect and most common and largest effect here doesn't show any association with birth weight. Those that are involved in beta cell function or a lipodystrophy-like phenotype, those are the ones that seem to show an association with birth weight. Whereas, for example, those involved in adult BMI or liver, there's no association there. So we hypothesize that those effects are mediated by insulin levels or either secretion or action. That's a fair assumption to make. But we want to, show that. So we have been carrying out a genome-wide association study of umbilical cord insulin and C-peptide levels. To date, kind of putting together all of the data we know about. Please shout if you have any cord insulin or GWAS data. We have about 10,000 mothers, 10,000 babies in the GWAS at the moment. That's not powered enough to identify individual variants associated. But what we begin to see when we actually add together we pool the SNP effects of SNPs known to be associated with fasting glucose levels. So the blue ones here are the maternal, the red are the fetal. These are the associations with umbilical cord insulin. We begin to see that those associated with higher glucose or lower insulin secretion are positively associated in the mother, negatively associated in the baby, and then taking those genetic variants that we know to be associated with a higher risk of type 2 diabetes genome-wide and lower birth weight genome-wide, we see the indications that the associations are in the direction we'd expect. But we're hoping to increase the power in that study. The lipodystrophy-like group of variants was actually something that I think we've only more recently begun to appreciate. So when the first studies of type 2 diabetes, GWAS of type 2 diabetes came out, it was clear there was a real big kind of beta cell function element. And we see those associated with birth weight. In the monogenic diabetes, we see those associations with kind of beta cell defects, show these big associations with birth weight and diabetes. In monogenic kind of lipodystrophy, there is less of a report of kind of big effects on birth weight, but we do see that there are that these variants which tend to associate with a higher metabolic risk and a higher adiposity, but also a lower metabolic risk, which various people have kind of identified. So examples here. With these metabolically favourable adiposity SNPs, we see that they are showing associations with birth weight. And this plot here is perhaps it's not displaying brilliantly, but what we have here on the x-axis is the SNP effects on BMI and SNP effects on birth weight for the fetal SNPs on the y-axis. And what's plotted are, in black and in yellow, are a couple of sets of variants that are known to associate with adult BMI. And what you can see with there is that the effects on birth weight are pretty heterogeneous. There isn't a kind of an association where kind of those with a large effect on adult BMI have a large effect on birth weight. Perhaps not surprisingly, because BMI is, there are lots of genes associated with appetite control. Those aren't having an effect in utero when the baby is kind of clamped to the mother's glucose levels. So we don't see a lot of good, strong correlation between the variants that affect adult BMI and variants that affect birth weight. But those that are associated with adipose tissue distribution, we do see more of an indication that they are having effects on birth weight. So the ones in blue are the metabolically favourable adiposity alleles, which associate with a favourable metabolic profile, but higher body fat percentage, higher ability to store adipose tissue subcutaneously versus ectopically. And those do tend to show an association, those with a higher effect on body fat percentage in adulthood, or correlates with a higher effect on birth weight. And so kind of a couple of things to finish off with. I wanted to kind of think about the birth weight variants as a group now. So all genetic variants that we found to associate with birth weight, how many of them are also kind of showing associations with type 2 diabetes, or how much of that kind of genetic component to birth weight that we've begun to characterize, how much of that is kind of indicating insulin-mediated growth, which I said is we think is accounting for about half of human birth weight. Well, actually, the answer is that overall, those genetic variants that we've found in the GWAS are mostly not associated with cord insulin levels. So this is some data from two different studies, a study of about 1,000 individuals from the Exeter Family Study, and then another 1,000 individuals from the much larger HAPO study. These are, on the left-hand side, the association between cord insulin or C-peptide levels. C-peptide is secreted in a 1-to-1 ratio with insulin, so it's an indicative of fetal insulin levels. They as we might expect, show a positive association with maternal fasting glucose levels in pregnancy. On the right-hand side is a fetal genetic score for birth weight, and then levels of cord insulin C-peptide. And as you can see, there's really no correlation there. Now, we know that the fetal genetic score and maternal glucose both are strongly positively associated with birth weight. And so what this is telling us, although we definitely have indications that several of the birth weight loci are associated with type 2 diabetes risk and show some indications of association with cord insulin, most of them are probably reflecting non-insulin mediated growth. And so just finally, I wanted to talk about the most recent GWAS that we've completed, which is a genome-wide association study of placental weight. And so we've been interested to look at the genetics of placental weight. We've got a preprint on medRxiv at the moment. If you want to find, I can't talk about it all here, but we found using 60,000 fathers, mothers, babies with information on placental weight, we found 40 regions of the genome that are associated with placental weight. We see a large overlap with the genetics of birth weight. So perhaps unsurprisingly, those genetic variants that tend to make a baby bigger are also associated with having a bigger placenta. We see that most of that genetic variation is fetal, although there is a maternal component. But what about genetics of placental weight and the relationship with insulin? Do we see similar associations as we do with birth weight? If we think about it, maternal glucose crosses the placenta, and that's available to the baby. Maternal insulin regulates the maternal glucose, but it doesn't cross the placenta. Fetal insulin is produced in response to maternal glucose, and we know that that makes the baby grow. But what happens with the placenta? Do we see similar associations? Well, when we look at when we add together the glucose-raising alleles, we see that in the mother, there are positive associations with placental weight and indications of opposite associations with placental weight. If we look at genetic variants that influence pancreatic beta cell function, we see that the fetal alleles are associated with higher placental weight and opposite effects in the mum. And then there's kind of indications perhaps less strongly with fasting insulin or insulin resistance. But these associations very much mirror what we see with birth weight. So the GWAS data do support a role for fetal insulin produced by the fetus in regulating placental weight and providing this link between fetal and placental growth. So just to summarise, the last 15 years of our GWAS studies on birth weight has seen a real progress in our understanding of the role of common genetic variation in birth weight. We now have 243 regions of the genome that are associated, and we've begun to separate the fetal from maternal effects and identify regions of the genome showing parent of origin effects. Fetal genetic variation in pancreatic beta cell function and adipose tissue distribution influences birth weight, and this contributes to the observational associations supporting the fetal insulin hypothesis. But importantly, not all type 2 diabetes alleles influence birth weight, and in fact, fetal birth weight alleles mostly appear to be associated with non-insulin mediated growth. And our recent GWAS results support placental growth being regulated by fetal insulin. And then finally, just to make the point, actually, as Anna alluded to earlier, our real priority is to increase diversity in our samples. The genetics of birth weight and placental weight are no different from a lot of all probably other human traits that have been studied in GWAS, being really, we've covered European ancestry populations so much better than others. So we're really keen to understand birth weight and related traits in different human samples and settings. And so I have lots of thank yous to make. I like to just flash up this slide just to show these are kind of the large numbers of people who contribute to these GWAS studies. And even just the group of us who meet every week at the moment to run the GWAS, the latest GWAS studies of birth weight and placental weight. These are the people that contribute, and it's a real pleasure being involved in such kind of dynamic and interesting consortia. And this is our team in Exeter. We have a PhD studentship available if anyone's interested, so look us up on Twitter. And thanks very much.
Thank you very much, Rachel. And so we have time for questions. And I have a question to start us off with. So the fact that most of the variants aren't explained by insulin-driven growth processes, but it seems they're rather, or to some extent, explained by body shape distribution and amount of fat. So if you were to adjust for the amount of for waist-hip ratio, basically body shape, how much does it attenuate the increased risk you see for type 2 diabetes in adults?
Oh, that's a good question. I think it's tricky. I think so. The adipose tissue distribution kind of variants have been identified in studies of adults, and we're kind of assuming that there's an effect on perhaps insulin resistance, but we don't really know the mechanism. It may well be that there's kind of more fat per se, so there's a higher birth weight, and that kind of relates to this metabolically favourable adiposity later. I think it's just an interesting thing that I think there's a lot more we need to understand before we can go further. Certainly, in terms of adjusting, it would be nice to have big enough studies where we can look at weight adjusted for length, so we can look at the fatness of a baby versus length, and I think there's lots to do there.
Great. We have two questions up here. Anna Gloin.
Thank you, Rachel. That was lovely. It's really good to see the new data. Thank you for sharing. I've got a couple of questions. The first one relates to signals, loci, where we know we have multiple signals. You showed us TCF7L2 as sort of sitting in the middle, not having an effect. And I just thought, well, that's more than one signal, and we think they're working in different tissues. So is there any scope to go back in and to sort of dissect that out, looking at whether it's working through adipose or whether it's working through islet, and perhaps then something might come out of the woods?
Yeah, I think we definitely need to do that. I think that would be a really good thing to do. I think talking of multiple signals, the KCNQ1 region, which you mentioned, is a really interesting one. We see a similar parent of origin effect on placental weight as we do with type 2 diabetes there, but there's another signal which seems to go more with birth weight than placental weight in the same region. Yeah, so I think there's a lot more kind of fine-grained work.
May I take the second quick question just to add on? This is ignorance on my part. You showed the total number of loci that you have for birth weight, and there were a large number that were associated with coming from the fetal genome, and then a smaller number from maternal genome. In terms of your expectation for the heritability, what's the sort of proportion that you expect to be able to explain from one or the other? And do we know that?
Yeah, I can't remember for sure what's expected or whether those studies were that clear, but certainly we see. Yeah, so I think with the fetal side of things, I think it's about 15% we've kind of explained from the SNP heritability in the fetus, and perhaps another kind of 5 or 6% from the mother. That I'm not sure what we would expect there, but certainly is holding up in what we see with the placental weight as well, which is, I guess, a fetal tissue.
We have a question up here and then a question down here.
Low birth weight protects against type 1 diabetes. Can you elaborate on this, the difference?
So yes, I'm less aware. I'm not an expert on the literature there. So I think there's a relationship between higher birth weight and risk of type 1 diabetes.
Low birth weight protects.
So yeah, and therefore the lower birth weight is protective. It's hard. I think it's really hard in observational studies to properly account for maternal glucose levels. So if there is a risk transmitted from the mother, then there may well be an exposure to higher glucose and a shared genetic risk, which would, so that therefore the higher birth weight babies higher risk, or lower birth weight lower risk. I'm not sure. We certainly haven't, because it's an autoimmune component, we haven't really looked at, we haven't really found kind of a link there. Some of the interesting one of the talks from this morning was about kind of a metabolic component of type 1 diabetes risk. So I think I'd be interested to look at that more and see whether that fits with our data. But I don't know for sure how much is kind of, how much you can separate correlation from causation, shared environment postnatally from genetics to really get at that.
I was wondering if one of these genes is imprinted at your study.
Yes, actually, several of the genes that showed parent of origin effects are imprinted. So we have associations with birth weight in the KCNQ1 region. Also, GNAS is another locus. DLK1. These are all imprinted genes which show some evidence of association with birth weight. Interestingly, some of the loci that show parent of origin effects are not in imprinted gene regions, and I'm not quite sure what that means, but there are certainly, and in fact, some of our analyses show that imprinted genes are overrepresented in those that show association with birth weight.
I have an additional question. Do individuals born with low birth weight progress differently to type 2 diabetes compared to individuals who are just at risk of type 2 diabetes?
I'm not sure about that. And of course, there is I think probably what we can't really do very well always is know whether a baby is kind of genetically small because they've got more kind of variants that are associated with lower adult height versus those who show growth restriction in utero due to kind of other external factors. And I think there would be very different kind of processes governing later health in those. So what we're concerned with is kind of normal variation in birth weight at term and less with the extremes.
Great, thank you. We have one last question up there.
Thank you for the exciting talk. My question in connection to birth weight that has been linked to a U-shaped association when it comes to type 2 diabetes, but also body trajectories from childhood to adulthood. They have an association that is different as compared to cardiovascular disease. Have you done any analysis in the connection between fetal growth and cardiovascular disease?
So yeah, actually, well, in cardiovascular disease, perhaps not, but certainly we've looked at relationship with blood pressure. So in terms of kind of that being a risk factor, we certainly see. So there is an observational relationship between lower birth weight and later higher blood pressure, and that's related to cardiovascular disease. So far, there has been some kind of disagreement in the evidence, but I think a consensus at the moment is that genetic variants in the mother, which raise blood pressure, are associated with reduced gestational duration, which is also associated with reduced birth weight, and that genetic risk is passed to the baby, which increases the risk later in life. So that, yeah, but actually we haven't looked at cardiovascular disease per se, or certainly we don't see the same kind of clarity as we do with type 2 diabetes variants.
Great. So thanks again, Rachel, for an inspiring talk, and please join me in thanking